Part 4, you reach the final part of the final exam review. Keep working hard, keep going, keep studying, keep fighting this good fight. You can do this, and we're gonna get through this together. Let's do this. All right, so here we have the organization of the nervous system, right? Our central nervous system, it's the brain and the spinal cord, that's it. The peripheral nervous system is everything else, right? So we're gonna have our sensory afferent division. This information is going to bring information to the central nervous system. Whenever it says afferent, that means information is going from the peripheral nervous system to the central nervous system. Then you have the motor / the efferent division. And whenever you see the word efferent, that means information is going from the central nervous system to the peripheral nervous system. So it's basically taking the information away from the central nervous system, whereas afferent is taking the information and bringing it to the central nervous system. So then you have your somatic nervous system and your autonomic nervous system. The somatic nervous system is all of the voluntary stuff. So somatic deals with skeletal muscle. The autonomic nervous system, that deals with involuntary muscle, such as cardiac and smooth muscle. And then this division breaks off even further, going into parasympathetic and sympathetic divisions. The parasympathetic division is rest and digest. Okay, so this is going to be when your body is relaxing. But the sympathetic division is going to be when your body is energized or when you're exercising, or it's also known as the fight or flight system. Picture you have to fight a mountain lion, or a mountain lion is right in front of you and it's hungry. What are you going to do? You're going to either fight that mountain lion to survive, or you're going to run away to survive, fight or flight. That's sympathetic nervous system. All right, so this is the neuroglia of the nervous system. So in the central nervous system, we have our astrocytes, right? And those astrocytes are the most abundant glial cell that there is. The role of the astrocytes, they help to support and anchor neurons to capillaries. That's one support. Another thing that they do is they clean up extracellular potassium. So these guys, they don't like extracellular potassium, so they're going to suck it in if they see that there's extra potassium hanging around. In addition, they're responsible for the recapture and the release of neurotransmitters. So they help to reuptake neurotransmitters back into the axon terminals of the neuron. And these astrocytes, they talk to each other through gap junctions, and they're able to communicate cellularly through these gap junctions. The next neuroglial cell is called microglia. And the microglia, you can think of it as like the mom of the nervous system, because microglia mom, the role of the microglia is to transform into macrophages to fight infections. Now, there's not really a lot of cells to fight infections in the central nervous system, especially in the brain. And so that's where microglia comes in and it fills that role. Then you have your ependymal cells. Your ependymal cells are located in between fluid-filled cavities. So let's say this is your spinal cord, and you have all your vertebrae right here. The tissue will be out here, and then you have your fluid-filled spinal cord cavity with all the fluid in there. The ependymal cells are going to line the ends of it. So ependymal cells will line those cavities. These ependymal cells have little cilia hairs on them, and that cilia helps to circulate the cerebrospinal fluid. The cerebrospinal fluid acts as a cushion to the brain and to the spinal cord, so they're able to absorb things like if you jump, for example, or move your head around, the cerebrospinal fluid protects your brain from hitting your skull. The cilia also helps to circulate dead fragments. So let's say a cell ruptures into cerebrospinal fluid. This cilia helps to circulate those dead fragments of that cell, so that if another cell like reuptakes the dead fragments, it won't be in too high of a concentration because of the circulation provided by the cilia. And then you have your oligodendrocytes. And these oligodendrocytes, they have multiple processes on them, and they can attach to multiple neurons. And their role is to generate myelin sheath on CNS neurons. Then moving over to the peripheral nervous system, we have our satellite cells. The satellite cells kind of encompass the cell body of peripheral neurons, and they function exactly the same as astrocytes, just in the peripheral nervous system. And then I put Schwann cells next to oligodendrocytes because Schwann cells build myelin sheath in peripheral nervous system neurons. And they also are important for the regeneration of peripheral neurons in the PNS. All right, so here's the anatomy of a neuron. All right, so first thing first, we're gonna start out with the first part of the neuron, the dendrites. So these spiky things are called dendrites. And these dendrites are where the neurotransmitters are going to bind to the dendrites. And specifically, they're going to bind to the ligand-gated channels located on these dendrites, whatever those ligand-gated channels may be. Maybe they're ligand-gated sodium channels, ligand-gated potassium channels, ligand-gated chloride channels. They're all going to be located on the dendrite, is receiving the neurotransmitter. And this little region right here, this is called the axon hillock. And the axon hillock is the area between the soma, the cell body, and the axon. So this is our cell body. We have our nucleus inside of there. And this entire portion right here is called the axon. The axon is where the action potential is going to be conducted. So this is the conducting portion of the neuron. And the AP is going to be conducted all along this axon and eventually hit the axon terminals. And there's two types of conduction, which we'll get into in a second. But here's our axon. And then this is where the axon terminals are. And so if you think back to the neuromuscular junction, you have all of these vesicles in the axon terminals that contain some form of neurotransmitter. Whether an EPSP or an IPSP, it depends, but some form of neurotransmitter is going to be inside of these vesicles in the axon terminals. And this is the secretory portion of the neuron. Now, one thing we should mention is that most neurons will have what is called myelin sheath. And this myelin sheath is produced by oligodendrocytes in the CNS and Schwann cells in the PNS. Okay. If a neuron has myelin sheath, we'll just write. If a neuron has myelin sheath, then the area between the myelins, this is called the nodes of Ranvier. Okay. Now, when we add another neuron to the picture, so when the action potential gets down to the axon terminals, they're going to release their neurotransmitters in the synaptic cleft, and then they're going to bind to the ligand-gated channels, whatever they may be, on the dendrites. In this case, the neuron before the synapse is called the presynaptic neuron. And the neuron that is being synapsed with and receiving the neurotransmitters is called the postsynaptic neuron. So that is the anatomy of the neuron. All right, so here are the structural and the functional classifications of neurons. Okay, so we have our multipolar neurons. They're going to have 3 or more processes, and these are going to account for over 99% of all neurons in our bodies. Most neurons will be multipolar neurons. Next, you have the bipolar neurons. The bipolar neurons have 2 processes that extend from the cell body. They're rarely found. They are very rare because they're only found in the retina and the nose, used for sight and used for smell. Then you have the unipolar neurons. The unipolars have a single short process. They're mostly ganglia in the PNS, and Unipolar neurons are going to consist of our sensory neurons in the body, which, as we can see over here, structural classifications of neurons. The sensory afferent neurons, these are the neurons that are bringing the impulse towards the CNS. All right, these sensory neurons are going to be unipolar, except for special senses such as sight and smell, where it'll be bipolar. But 99% of the time, sensory afferent neurons are going to be unipolar. And they're found in the cell bodies in ganglia outside of the CNS. And then next we have the motor efferent neurons. These neurons are going to bring the impulses away from the CNS to the effectors to cause the intended action to happen. These neurons are multipolar, and the cell bodies of them are located in the CNS. And then we have our interneurons, which are the neurons that are between the sensory and the motor neuron. And these interneurons are going to be multipolar. So here's a visual of where you find the sensory and the motor neurons. Sensory neurons are to come in synapse with interneurons, which are multipolar, in the spinal cord, in the CNS. So as you can see, the sensory afferent neurons, afferent, bring information to spinal cord, to CNS. We have our interneuron, which is multipolar. And then we have our motor neuron, which is multipolar. And the motor neurons bring information away from the CNS, which is why they are efferent neurons. All right, so now we're going to talk about the generation of an action potential. We're going to talk about resting membrane potential, what that's look like, how that's established, and then we're going to go through each phase of how an action potential is generated. So each phase of this graph we're going to go over. So starting out with phase one, on our cells we have voltage-gated sodium channels and voltage-gated potassium channels. Now just to review on voltage-gated channels, if the channel is voltage-gated, it means that it's going to open because of a change in membrane potential. More specifically, it's going to open when threshold is reached on the action potential graph. And as you can see on the voltage-gated sodium channels, we have what's called an activation gate, and we have what's called an inactivation gate. The voltage-gated potassium channels, they only have one gate, but the voltage-gated sodium channels, they have two gates. The activation gate in the starting phase at resting membrane potential, so at phase one, the activation gate is going to be closed. The inactivation gate is going to be open. So this is what both channels look like at resting membrane potential. And this is what is going on behind the scenes 24/7, 365. We have our sodium potassium pumps, okay? These sodium potassium pumps use ATP, meaning they use active transport, and they're pumping ions into an area where there's already a lot of them. In other words, there's already a high concentration of these ions where the sodium potassium pump moves them to, okay? So outside of the cell, there's a high concentration of sodium, meaning there's a lot of sodium ions in the extracellular space of a cell. In the inside of the cell, there's a high concentration of potassium ions, meaning that there's a lot of potassium ions inside of our cells. The sodium potassium pump takes 3 sodiums from inside the cell and moves them outside the cell into an area where there's already a lot of those ions, those sodium ions. So we can say that the sodium potassium pump moves sodium ions up/against their concentration gradients. Then it takes 2 potassium ions and it pumps it down in the intracellular space of the cell where there's already a high concentration of potassium ions. And in other words, there's already a lot of potassium ions in the area that the potassium ions are moving to. So those also pump up/against their concentration gradients. And whenever you pump up/against a concentration gradient, it's called active transport, which requires energy in the form of ATP. So that's how the sodium potassium pump works. And the sodium potassium pump establishes resting membrane potential. you have your leakage channels, sodium leakage channels and potassium leakage channels. And these channels passively diffuse ions down/towards their concentration gradients in an area where there's not a lot of them, right? So sodium moves down, there's not a lot of sodiums inside, not a lot of potassiums outside. They passively diffuse ions. And when you passively diffuse ions, it means that you're moving down/towards their concentration gradients, okay? Another thing to note here is that there's more potassium leakage channels than sodium leakage channels, and this is because cell membranes are more permeable to potassium ions. These leakage channels, they help establish the resting membrane potential. In other words, they aid the sodium-potassium pump in establishing resting membrane potential of -70 millivolts. But they don't establish the RMP themselves. That is the job of the sodium-potassium pump. They help the pump, but they don't establish RMP themselves. So if all of this makes sense to you, then we're going to move on to step 1.5. All right. So in step 1.5, we see that we have depolarization going on right before threshold. Okay. So when we build up to threshold here, we need to open up ligand-gated sodium channels on the neuron's dendrites. So this is the presynaptic neuron, this is the postsynaptic neuron. And remember, we have all the vesicles in the axon terminals of the presynaptic, releasing it into the synaptic cleft, binding with, in this case, since it's depolarizing, ligand-gated sodium channels. This opens up those ligand-gated sodium channels, allowing for sodium to come in, to depolarize the membrane up until we hit -55 threshold. Once we hit threshold, then we move on to step 2. Now, since we have hit threshold, voltage-gated sodium channels are going to open up, and they're going to open up by the inactivation gate opening the channels, allowing for sodium to influx the cell, causing further depolarization of the membrane. Then in step 3, the voltage-gated sodium channels, so step 3 starts from the spike down, and in step 3, the voltage-gated sodium channels inactivation gate is going to plug the voltage-gated sodium channels, preventing any sodium from coming in. Once that happens, voltage-gated potassium channels are going to open up, and they're going to efflux. Potassium's going to leave the cell, causing the membrane to get more negative again. Now in step 4, this is when the voltage-gated sodium channels reset back to their OG positions. Okay, so the activation gate closes up, the inactivation gate peels off, and now they're back to the OG position. And the voltage-gated potassium channels are still effluxing potassium out because voltage-gated potassium channels take longer to completely close up. So these guys are still open for a little while longer, which is why we have hyperpolarization. Then eventually they're going to close up. And then all of this is still happening in the background. So once the cell is at rest, the sodium-potassium pump reestablishes the resting membrane potential, and then all the leakage channel stuff is going on in the background. So phase 1 of the graph is resting membrane potential. Phase 2 is depolarization. Phase 3 is repolarization. And phase 4 is hyperpolarization. And so that is how an action potential is generated, and this is how resting membrane potential is established. So this is a good graph that I just thought I would show you that relates to temporal summation. So we can see here is that if we have a stronger stimulus, as we can see right here with the buildup, that means that the more frequently APs are being generated. So we increase the frequency of APs. All right, so now we're going to talk about the absolute refractory period and the relative refractory period. So just kind of go over what we just talked about. All of these that you see in red here are voltage-gated sodium channels. And this is what they're going to look like at each phase of the graph, right? We know at resting memory potential, the activation gate is going to be closed, inactivation gate is open. Once we get to the depolarization phase, both activation gate and inactivation gate are open. During repolarization, the voltage-gated sodium channels are inactivated, with the inactivation gate closed and the activation gate still open. And then once we hit back to resting memory potential, we return the voltage-gated sodium channels to their OG positions. The absolute refractory period is when it is impossible to generate another action potential. Okay, and it is impossible to do so because, so you see it occurs during depolarization and repolarization. During depolarization, it's still, the channels are open and sodium is coming in through these channels. So you already have an action potential going. And then during the repolarization phase, the voltage-gated sodium channels are in their inactivated state with the inactivation gate plugging them so that no sodium can enter anymore. The relative refractory period is where it is harder to generate an action potential, but it is still possible to generate one. But you're going to need a very strong stimulus to generate one for two reasons. One, because during hyperpolarization, we have some voltage-gated channels that are back to their original position, ready to receive another action potential to open their activation gates back up. But the thing is this process of resetting them back to their original positions happens very gradually, right? So during the hyperpolarization phase, some voltage-gated sodium channels are still going to be resetting. Some are still in their inactivation state, as you can see up here. And when they're in their inactivation state, they cannot be opened up. The inactivation gate has to be off and the activation gate has to be closed in order for sodium to come down, open this up, and depolarize. The second reason why it is harder to generate, but still possible to generate an action potential, is because in the background of all this, we still have voltage-gated potassium channels that take forever to close. So they're still open and potassium is still effluxing, which gets us hyperpolarized, making it harder and making the journey to threshold a lot longer. So compare that with only a couple of voltage-gated sodium channels ready to go, and many of them still in their inactivation state. You're going to need a very, very strong stimulus because you got only a few voltage-gated sodium channels to work with. And then once we get back to RMP, all the channels are back into their original positions, ready to fire again. So these are EPSPs and IPSPs. With an EPSP, our presynaptic neuron is going to release the neurotransmitter of glutamate or ACH that are inside these vesicles, right? And we know that in the background, we got the voltage-gated calcium channels opening up, and the calcium comes in, and it releases the glutamate or ACH into the neurotransmitter, and it binds with ligand-gated sodium channels. And so therefore, sodium comes in to the postsynaptic neuron, and it causes the build of the graded potential. So it causes depolarization in order to hit threshold. And the summation of EPSPs makes this possible. So one EPSP won't get you to threshold. It'll only get you like a little high like that. You need multiple EPSPs to get you to build up to threshold, and then you hit threshold and you fire that action potential, and those voltage-gated sodium channels open up. Well, EPSPs stand for excitatory postsynaptic potential. IPSPs stand for inhibitory postsynaptic potential. And the goal of the IPSP is to stop action potentials from being firing by hyperpolarizing the membrane. So with an IPSP neurotransmitter will either be GABA or glycine, okay? And again, you have all the background voltage gated calcium channels, calcium coming in, all that stuff going on in the background. You release the neurotransmitter into the synaptic cleft, which is either one of these two, and they're going to bind to either ligand-gated chloride channels or ligand-gated potassium channels. If they bind to ligand-gated chloride channels, chloride is a negative ion. So that's going to come in and it's going to hyperpolarize the membrane. If it binds to ligand-gated potassium channels, potassium is going to exit the postsynaptic neuron, which makes the membrane potential more negative, therefore hyperpolarizing it. So it's the summation of IPSPs that prevents an action potential from being fired. And if you take a look at this graph here, one IPSP will hyperpolarize you, one EPSP will depolarize you, but if you take one and the other and combine it together, they cancel each other out. So here's continuous conduction and saltatory conduction. In continuous conduction, there's no myelin sheath on the neurons, so you're going to continuously generate the action potential over and over and over again on every spot of the axon. So you're going to go through all of the action potential generation steps that we talked about earlier in this video. But whereas in saltatory conduction, the difference is that there's myelin sheath on the neurons. And the myelin sheath does not allow for leakage channels. So therefore, the voltage doesn't decay along the axon as much. And the myelin sheath basically speeds up the action potential propagation down the axon. And then it's regenerated only at these spaces, which are called the nodes of Ranvier. Therefore, saltatory conduction is going to be way faster in terms of action potential propagation speed compared to continuous conduction, because here you have to continuously generate it, whereas here myelin does not allow for leakage channels, so you won't decay the voltage and it will just go down the axon faster.